Okay, so let us start with the second lecture on this regulatory aspects of drug development. And as we explained, we will be mostly focusing on the biologics side of drug development. So, today I am going to introduce you to biologics, what is biologics, but let us start with simple drug discovery. This kind of picture you might have seen earlier as well, drug discovery and development timeline, how much it takes to develop one drug, the time wise, money wise, and what it all involves. So, if you start like a drug discovery program, discovery is like you are going to find a new drug for a certain indication, right. For a particular disease, you are going to find a new drug. Now, these new drugs starts from millions of or at least 10,000 of compound. That initial part is discovery program. So, discovery program it can starts with now a biophysical or a docking or bioinformatics. You find have a different kind of molecules and you know a target, right. All the drugs are developed against a target. Target can be protein, target can be nucleic acid, target can be anything, any biomolecule which you are targeting and trying to find one molecule that binds. So, starting with 10,000 molecules or 1,000,000 molecules that shows the binding, right. So, binding to this particular kind of molecule. So, this particular kind of molecule, how do you know whether it is binding or not? First you start with a easy way is to start with a computational tool, because पैसा उसमें कम लगता है, right. So, start with simple docking. Suppose you are finding a drug for HDAC, right, HDAC inhibitor. What is HDAC? नाम सुना है? Any of you? HDAC, histone deacetylase. So, these are probably going to be used for cancer treatment. So, if you want to find a drug against HDAC, how are you going to start? First, you are going to have a structure, high resolution structure of HDAC. Then you know where is the binding pocket of this HDAC, that can be again done computationally. Now, you have seen in computer what is the binding pocket, and then you can do pharmacophore mapping, find a molecule that will fit to there. So, you start with some defined knowledge in this domain that this pocket looks like a particular arrangement and these moiety can fit it here. So, in computational you can using computational tool you can design your molecule that will fit to this pocket. So, that is a, or you can just randomly screen it using any biophysical tool. You can screen the molecule and see which is binding, right. We can in MSc we have done the practical, you looking at the fluorescence spectroscopy and seeing if the fluorescence of this molecule is changing upon binding. That is a discovery program. So, that discovery program takes say long time. You identify few molecules that at least shows good binding. Good binding means micromolar kind of binding to this my particular target. Otherwise, you modify the compound, you make it better and all those. So, those part is initially done in R&D setup, research and development setup, where you find a molecule that shows efficacy to the binding to these molecules of your choice. Now, once you find a molecule, what is the next step? In vitro analysis, that is what you can do. Even in cell analysis you can do, take these molecules. So, if you find a inhibitor or something, say mitotic checkpoint inhibitor, you check in the cell whether it is working or not. That again comes into say R&D kind of setup. So, you start doing that kind of setup. Now, after that you move to preclinical setup, like you look at the molecule in mouse model, rat model, rabbit model, any of these preclinical model, and look at the ADME property that you might have studied in various course, whether it is getting absorbed, distributed, metabolized and excreted from the system. So, this much you can do in a preclinical setup, right. Its toxicity, efficacy, many of these things can be done essentially in the preclinical setup. So, suppose you started with 10,000 1 lakh compound, you end up with 250 compound. Those 250 compounds, some of them will pass to whatever criteria that you have set up, then some of will not pass, and then out of that you can get say 5 compound or so. Then you are going to do clinical trials of those 5 compound against any indication that we are developing it. So, in clinical trials it may involve human trial. You take a permission that it is not toxic, it is safe, and it is doing whatever job it has. So, 3 phase of clinical trial that I will come in a moment. What are those 3 phase? So, clinical trial you end up with a 5 compound set, and then finally, if you get these compounds working at least 1 or so molecule, then you submit all your data to any regulatory agency or approval agency, like in USA it is called FDA, right. So, FDA, they thoroughly review all your data, look at what you have developed, and then finally after all these years of research, say about 13-14 years of research or 15 years of research, one molecule gets approved and then it can be marketed. So, if you to bring a new molecule to the market, about 12 to 15 years and about 2.6 to 3 billion US dollar is required. So, this making a new drug is more expensive than sending a satellite on moon on Mars, right, and lots of effort which goes behind the carpet to bring a new drug. That's what it is demanding and required, right. So, drug approval process, that's what, so we said that it has to be approved by some agency. So, prescribed drug comes under this at least in all the developed country, and India is also making its own policy and FDA. So, all new drugs or whatever drug is developed, they have to pass through a regulatory process. So, prescription drugs are regulated under the Act in USA, at least Federal Food and Drug Cosmetic Act 1938. उस समय तो हम लोग गुलाम ही थे, तो अपना कोई ड्रग वाला था नहीं, but still we follow lots of stuff that has come from USA, EMA, and India is also developing its CDSCO, is also becoming more stringent in regulating the market. So, only about 1 of 5,000 potential drugs successful pass through the testing process that is approved by the FDA for patient use, right. So, when we go into actual case study, you will see how difficult it is and how many companies went bankrupt chasing behind a new drug. We will discuss all the failures and successes. But before I really venture into this course, let us understand the language of this course. So, some terminology I will explain to you. Many of these you will be aware about. So, say a pharmaceutical product. How do you define a pharmaceutical product? Right? We use some medicine. So, typically it is a chemical drug. Pharmaceutical product, aspirin, it is a chemical drug that you take, consume, and then it creates a therapeutic effect. Sar dard ho raha hai, you take it medicine, it creates some therapeutic effect. That's one can say it's a pharmaceutical product. So, it may include this treatment of your, or it can even like some kind of prevention of symptoms of illness or injury in human or animal, those will be called pharmaceutical products. The term drug, like commonly we use drug in good sense, not in bad sense. Bad sense also has similar effect, but a drug sense applies to any pharmaceutical product that is used for diagnosis, treatment, prevention of any indication that we call. A new drug, what we discussed, that new drug takes time, right. So, new drug जिसका effectiveness has not been shown previously, that will be called a new drug that we are say most of the time people discover. The existing medicines are there, but a new drug against a new disease, that will be called a new drug whose effectiveness or where it is going to be used has not been discussed, right. Now, these are all established drug. A generic drug, right, most of the time we have heard about generic drug. Generic drug are those drugs who are already developed by someone, its active pharmaceutical ingredient is known, but some other company after the expiry of patent makes it in a prescribed format, that agency has given a prescribed format. According to that, one can make a generic drug, right. It will be used for the similar kind of indication whatever RLD reference listed drug is there, and these drugs are like they should be matching with the like a reference listed drug in terms of dosage, in terms of the safety, the strength and other things. Now, another two important terminology you will encounter, it is called NME and NCE. So, NME is new molecular entity, right. So, it is an active ingredient of any drug that has been never marketed, at least in US, that is what, and NCE is new chemical entity that has not been marketed. So, those are for a small molecule based drug. Yes, go ahead.
The generic drugs can only be sold in market after the expiry of this patent of new drug.
Yes, generic drug can be only sold in the market after the expiry of the patented drug. So, many of the drug goes out of patent after how many years? Some drugs has 12 years, some has 12 to 15 years. So, typically drug has a life cycle of 12 years. After that, if you match the criteria, you can make and sell the drug. So, in India, most of the companies we have we are so called pharma hub, right. But in this pharma hub, most of the drugs that are manufactured are generic drug. So, as I said, the main thing is here, the amount that goes is huge for getting a new drug, but making a generic drug amount is not that much, because the molecules, the API is known, the molecule which is effective is known, the guidelines are more or less set ki isme itna milana hai and you can make a drug, but still lots of things has to be done, it is not that straightforward. Okay. But the good part of making generic drug, that affordability increases, right. So, suppose you are getting a headache, a simple headache, right, and if you are buying 100 rupees ka drug, you will think twice to buy a drug, or this is even for simple disease I am saying. But if it is 1 rupees or 50 paisa drug, anyone can buy and use it and get rid of these problems. And even for the chronic disease, you see, before the generic market opened, the drugs were so costly, and how many people can afford this. So, generic drug opened a new avenue to democratize or to reach out to the common people, and that is a big advantage that happened after the market has been opened for generic manufacturers. Now, coming about biological products. So, therapeutic biological products are basically they are coming from the live organism. So, it coming from various kind of organism, it is a protein based drug coming from the living organism, and they are used to treat, cure disease, right, for various things. So, all those which comes from the live organism, like a protein or like a monoclonal antibody or like a urine product or like a blood products, many of these we can call it a biological product in a broader sense. Now, biopharmaceutical, another term, biopharmaceutical, specific type of a drug that has been created via genetic engineering of an organism. What I mean by genetic engineering of this organism? In our lab, we always overexpress a protein, right. Why do we overexpress? get more yield, overexpress where? In bacteria. Why? Because the protein related to human does not belong to bacteria, like. So, what we do? Heterologous expression. Human ka protein we clone in a bacterial gene and overexpress a protein, so that we get enough protein. That is what just basically in biopharmaceutical we also do. Take a drug or a protein drug molecule that will be used for human application, but overexpress in any other system, heterologous expression system. It can be bacteria, it can be yeast, it can be insect, it can be human cell, and you overexpress to get enough quality, because now we are making a drug not in a small amount, you are manufacturing at large scale. So, those will be whatever is expressed in other organism by use of genetic engineering will be called biopharmaceutical agent or biopharmaceutical product, and this biologics, biopharmaceuticals broadly we classify into that. And biosimilar is a generic version of biopharmaceutical product. Okay. So, it is a FDA licensed product and you make similar to whatever was already marketed, like insulin has been made by say Eli Lilly and Biocon is making insulin. So, insulin molecule is known, now patent is expired, now some other company is making same insulin using its own, like keeping the insulin same, but other constituents can vary. It should be structurally same, effectiveness should be also same, and if that is matched, that will be called biosimilar of the reference listed drug. So, biopharmaceutical, biologics, vaccine, protein, tissue, these are the things. So, now we know this common term that we are going to use: pharmaceutical product, drug, generic product, biologics and biopharmaceutical. We will move ahead. So, we are going to design a car. How are we going to do that? Designing a car, like developing a drug, as I said, more difficult than making a satellite to send it to moon. So, I am just starting with designing a car. So, first you make various design, right, various design on paper, you make on paper. So, first stability, look wise and all those, how it looks. So, first we make on paper, then we try to make it on models, then parts are manufactured and finally parts are assembled in some assembly line, and then it is tested for robustness, effectiveness, safety, and then it is launched in some whatever auto expo. So, similarly and there are many manufacturers that makes. So, similarly biological drugs, as I said, follows various kind of process. First, discovery, whether it is small molecule based drug or a biologics drug, the first and most time consuming and effort consuming process is discovery phase. And discovery phase comes because not one day somebody gets in dream कि ये molecule काम करेगा, lots of failures, continuum of learning, and then you confirm a process to discover something new. So, discovery takes lots of effort. It can come from some knowledge, like as last class we were discussing about insulin. So, insulin first isolated from the pig, injected to the human, and it shows effectiveness. So, people thought ki insulin co treatment, then I said right, it can help us. So, discovery is very important phenomena in the drug discovery. So, first you discover a molecule that shows effectiveness, right, and then you do pharmacology, find the effect of this where it is happening. So, mechanism of action you have to do. So, any molecule that we discover first in vitro, then in cell, is it showing the mechanism? Say let us start with insulin. Suppose I design an insulin molecule, how do I test whether it is active or not? Whether it is active or not, whether it is doing something, some action or not, how we can do that? At least in the discovery phase or R&D setup or in our lab? You check on cell. What do you check?
First we start toxicity.
The toxicity comes later. First, mechanism of action you have to establish, right? Mechanism of action. Insulin का क्या काम है? It should bind insulin receptor. So, say I take a cell which has insulin receptor expressed and I develop an assay which will show the action when insulin binds to this receptor, isn't it? So, that will tell us that whether I have got right kind of insulin or not, which is binding to insulin receptor giving me signal. If that is happening, then again GLUT4 receptor and all those glucose will be taken up, glucose metabolism can trigger. So, first you have to, in the discovery phase, you have to develop a mechanism of action. Now, if that is robustly identified, what is the next step to understand the PK/PD? What is PK and PD? Pharmacokinetic. What is the difference between pharmacokinetic and pharmacodynamics?
So one is how drug is affecting the body and the other is how the body is metabolizing the drug.
Very nice. So, this can be tested at least in the preclinical model, right. So, because you know doing a new investigational drug on human is difficult. So, this kind of study you can do in preclinical model and you can collect the serum of the rat or mice, analyze where drug went, you can look at the effectiveness, whether it has damaged any organ or not. So, this pharmacokinetic, pharmacodynamic comes, and similar can be add with a toxicity model. How much toxicity this drug is creating, is it safe for use or not? The safety or any adverse event has to be accounted for that. Now, once the toxicity and in a preclinical setup happens, then it goes to phase 1, phase 2 and phase 3 trial. So, first tolerance to dose exposure and response, right. So, small dose on selected group of people will be given, that first it should not cause any adverse effect, people are able to tolerate that small doses and all those. Then in phase 2 some larger pool will be taken and you look at the therapeutic effect or efficacy and safety of this drug, and finally then result will compared with any standard care that has been developed by any methods. So, once this is done, then we are already set to do. So, as I mentioned, the first step is done in R&D lab, where you develop an API stands for active pharmaceutical ingredient, any molecule that is effective, like rest other are adjuvant or whatever. So, first most important thing is discovering the active pharmaceutical excipients or adjuvant or whatever you call, right. जैसे हम एक टैबलेट खाते हैं या कैप्सूल खाते हैं, उसमें एक एक्टिव फार्मास्यूटिकल इंग्रेडिएंट होता है, बट वो उसका कॉन्सेंट्रेशन इतना होता है कि उसको खाना या ऐसे मेजर करना इज डिफिकल्ट. सो इट इज मिक्स्ड विथ मेनी अदर थिंग्स विच आर टिपिकली न्यूट्रल टू योर बॉडी एंड देन दैट ड्रग इज फॉर्मुलेटेड. So, that is called excipients, adjuvant or whatever. So that's the main thing is active pharmaceutical ingredient that has been discovered in our R&D setup. Okay. Now, that actually is that shows the real potential and drug moves into next developmental stage. So, first these all are done in R&D setup. R&D setup typical like our lab, they have all the biophysical, biochemical, cell based assay. You develop a new drug, new API in R&D setup, and then you move to next stage and where you like whatever preclinical study if you are doing. So, early stage of development involves preclinical testing that is done in small organism, at least rat, mice and all those you have, and that carries in an animal testing facility for its toxicity and all those. So, if it is successful, so finally you reach to any organization like regulatory organization and you inform that I am developing and a drug against this indication, this is my active pharmaceutical ingredient, this is my R&D data and this is my preclinical data. You submit all those dose here. Why you want to submit it? Because preclinical के बाद क्या होगा? क्लिनिकल. क्लिनिकल किस पर होगा? ह्यूमन पर. तो अब तुम किसी ऐसे ही ह्यूमन में जाके अपने मॉलिक्यूल के अपना मॉलिक्यूल तो इंजेक्ट नहीं कर सकते. दैट इज लॉट्स ऑफ इथिकल इशू, लॉट्स ऑफ सोशल इशू, लॉट्स ऑफ इशू. तुम अपने ड्रीम के लिए किसी के शरीर में कोई भी चीज तो नहीं डाल सकते ना. सो इट हैज टू बी अप्रूव्ड बाय कॉम्पिटेंट ऑथोरिटी that now you can go and do it, but before you do, you submit all those investigational new drug application INDA, okay, to FDA and request that I need permission to begin the trials on human. So now you look at the regulatory agencies are engaged from early days of a drug development. Preclinical करने के बाद you are submitting all your data and they are essentially scrutinizing all of your data to allow you, now you are ready to do preclinical test. Okay. Now here comes lot more engagement of various stakeholders who are part of this drug development program. So let's understand who are the part of drug development program. I will narrate a simple my own experience with little bit that I wanted to do with human. I wanted to do some research related to diabetes using human serum sample and I collaborated with KEM hospital. बोला मुझे एक्स्ट्रा ब्लड सीरम नहीं चाहिए, आप लोग जो लेते हो ना उसी में से थोड़ा 1 ml मुझे भी दे दो. That's the my intention was, but that has to be approved by ethics committee because now we are doing some research on ethics committee. Now ethics committee consists of various people, डॉक्टर हैं, साइंटिस्ट हैं, सोशल साइंटिस्ट हैं, इकोनॉमिस्ट हैं, मैनेजमेंट वाले, पता नहीं कौन 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 है. And I look as a scientist point of view, what I wanted, serum to do some research. For me it was straightforward. मैं तो यूज किया हुआ ब्लड ले रहा हूँ किसी का, मैं like I am not taking anything which is, but their perspective is different. So lots of review process happens to my application, 8 rounds of revision, lots of changes, lots of questionnaires, lots of declaration. Finally I got approval, but for taking drug we have to pay some small token as a compensation because they use their fare or whatever to come. So you can think of this much ethical clearance required for any research that you do with a human sample. But what happens if you are going for clinical trial? So clinical trial, all this way, preclinical, you have some data collected, you fit that data. If it looks positive, you go to phase 1 where the small doses are given on a small number of people, and that is also given, and then preclinical phase 1, phase 2 and finally registration. So rigorous early phase program assessment and priority is essential for setting the finite resources. So that is kind of a patient friendly and finance friendly as well. Because finance friendly, why I am saying, each clinical trial that you are doing has to do with safety, efficacy and all those. So you are buying the insurance for that person, you will be responsible, any adverse effect can come, and drug development company. So typically this drug development company, they join hand because you think of the money that goes in. फेल होने की संभावना कभी भी है. हमने चूहों में दिखा दिया, आदमी में काम करेगा या नहीं, right? There are many failure story that we will discuss with you. So if a drug has worked in our lab, उसका कोई मतलब नहीं है. खुश हो जाओ पेपर पब्लिश करके, साइटेशन बढ़ाओ. फर्स्ट, दिस इज द बिगिनिंग स्टेप. This is the beginning step of your drug development program. You have to next, like in biophysics, you have to take it to the cell level, you have to take it to the animal model. If even in animal model it has been shown your efficacy, at least you have some hope that we can go. But money involved in clinical trials is humongous. Is humongous because you have to do the various clinical trials. I will just share some information since this is an advanced level course. You know why India did not allow Pfizer and Moderna vaccine to be sold in India? Any one of you know? It was for emergency. All the vaccines were given the approval for emergency use only, right?
mRNA vaccines didn't pass the ethics committee.
That's right. It did not pass through ethics committee. So first of all, none of the clinical trial were done in India. First and most important part. So they wanted that these vaccines has to be tested in India or Indian population, then only we are going to approve you. These companies said, ना हमने कर लिया है, आपके यहाँ कोई हम टेस्ट नहीं करेंगे. और they wanted some kind of assurance from the government कि हमारे वैक्सीन से कुछ लोग मर गए तो जिम्मेदारी मेरी नहीं आपकी. भारत सरकार ने बोला भैया अपना वैक्सीन तुम अपने पास रखो, हम जो बनाए हैं उससे हम खुश हैं. 2 of our vaccines developed in India by Bharat Biotech, ICM with ICMR. किसने ये क्या है अभी जो मूवी आई थी द वैक्सीन वार, किसने देखा? वैक्सीन वार इज ए नाइस वॉच मूवी. इसमें थोड़ा बहुत मसाला हो सकता है क्योंकि मूवी है फाइनली, बट इट्स ए नाइस मेड मूवी व्हाट एक्चुअली टेक्स to make a vaccine and how much scientists devote their energy time to make such kind of biological drug. So vaccine war is a must watch movie. और वैक्सीन या कोई भी ड्रग बनाने के पीछे इकोनॉमिक्स का कितना बड़ा हाथ है ये भी उसमें थोड़ी बहुत दिखाया गया है. सो इकोनॉमिक्स इज ऑल्सो इन्वॉल्व. बट so first of all it was they said they are not going to test in movie. Those are mRNA vaccine as you mentioned. mRNA vaccine के लिए cold temperature पे स्टोर करना पड़ेगा. भारत में कहाँ कोल्ड टेम्परेचर की फैसिलिटी है? मुंबई, दिल्ली, जयपुर, ऐसे ही बड़े बड़े शहर, बेंगलोर, हैदराबाद. बट मोतिहारी में बिजली ही नहीं आती है, वहाँ पर कैसे -80 मेंटेन करोगे? कोविड तो उधर भी हो रहा है. इट विल नॉट वर्क. और हमारी वैक्सीन कैसे कैसे डिस्ट्रीब्यूट हुआ, ऑल ऑफ यू माइट हैव सीन इट. आइसक्रीम वाले डब्बे में 4 डिग्री पर मेंटेन करके वैक्सीन गया एंड देन दे यूज्ड इट. So these are the things. Therefore, that's what the whole intention of taking this course is. You need a local solution for a global problem. It cannot be just imported. So it has to be tested thoroughly, and all the vaccines approval are given on emergency use. I know and I fully understand and I am aware that all the detailed testing has not been done on even these vaccines. But those were the demand of the time and lots of, as I said, here it is a continuum knowledge. So here continuum of learn and confirm process. So our Covaxin was made on the basis of that, and therefore even if some tests were like were not published in public domain, they finally published in Lancet, but they had the data and using that data CDSCO decided to approve it. So once these preclinical studies are done, now it passes, clinical study is successfully done, then the companies can submit. Now this is called new drug application to FDA. This is called NDA, new drug application. इससे पहले क्या एप्लीकेशन उसने फाइल किया था प्रीक्लिनिकल के बाद? INDA, investigational new drug application. Now after clinical trial it is NDA, new drug, and if that is approved, now company can sell the drug, but this drug has to be monitored. क्योंकि हमने मानो क्लिनिकल ट्रायल तो like we did it on few thousand people, and they did not show much effect, the efficacy was there, but you do not know, right, whether what will happen to mass. So, many times drugs passed in clinical trial, but later on it creates a problem and drug has been recalled it. So, then comes again role of regulatory agency, FDA and all those. This one can call it pharmacovigilance or looking at the adverse effect. And if you remember, if you go on our COVID app, when vaccine was being administered, so they were putting various kind of data. कितने लोगों को वैक्सीन दिया गया, कितने लोगों को एडवर्स इफेक्ट हुआ, बिकॉज दैट इज प्रोबेबल. वैक्सीन का या कोई भी ड्रग का एक पर्सन में एक इफेक्ट हो सकता है, दूसरे पर्सन में एडवर्स इफेक्ट हो सकता है. सो दैट हैज टू बी स्टैटिस्टिकली वेरीफाइड दैट इट इज not a problem. So, to summarize what we have learned till now, in a drug development process, starting from R&D finding an API, it has to be done a preclinical study where you monitor the efficacy of these drugs on model organism, then phase 1 clinical test of safety of these drugs on a small patient size like a 20 to 80, and then clinical test has to be done in phase 2. Number of patients can be few dozen to few hundred, approximately 30% moves to phase 3, and then in phase 3 clinical trial one can do of efficacy of these, number of patients can be 1000 up to 1000, and then you find a new drug application, and then this drug is manufactured, licensed and sold in the market, and finally FDA results is taken, and finally this drug efficacy, if everything goes fine, then it is used. Okay. So, with this background, what are we going to do in this course? We are going to understand and try to do as it was discussed by Naren. In mind, you are going to develop a drug. That is one of the important way of testing you. So, about 9-10 people are there, we will divide into 2 groups. You will be developing, one group can develop one drug, another can be another drug, and that starts from the beginning. So, your projects start now. You think, you make a group and develop your own drug, and at the end of the day your evaluation will be done on what drug you have developed, what kind of experiments you have thought, what will be outcome, and how you are going to present to FDA, and the other team will be acting as a FDA, and role can change whether they would like to approve your drug or not. Okay. So, that is the way we are going to look at. So, we will be starting with design of vaccine and biologics, the understanding the normal distribution, design of experiment, right, pharmacology, toxicology, interference and statistical significance, some ANOVA test and all those we can learn, process development, manufacturing, process capability, process stability, because finally we are going to scale it up, and clinical trials, regulatory approvals, and then specification, and finally risk assessment and summary and conclusion. That is what we are going to do in this. Okay. So, developing drug for what? Betterment of the society, whether it is human, plant or animal. So, in say if we are developing for human, there are some 27,000 gene and there are so many proteins, and they are essentially potential target. So, one can develop for various category, right. So, these are like a cell adhesion protein, structural protein of muscle, motor, ion channel, like many of the drugs are ion channel blocker. So, you have to find a target against which you are going to develop a particular kind of drug. Channel blocker कहाँ use करते हैं? Ion channel blocker. Various पेन में यूज करते हैं और and something related to even your blood pressure and all the channel. So, these are the targets, these are target protein, because we are going to develop a drug against a target. So, there are various targets, receptors, kinases. अभी मैंने नाम बोला था HDAC का, उसी तरह कई सारे काइनेज हैं. काइनेज का क्या काम होता है? फॉस्फेट डालना, राइट, किसी मॉलिक्यूल पर. काइनेज का ब्लॉकर हो सकता है. तो दीज आर वेरियस टारगेट against which we are going to develop a drug. Since we are going to mostly restrict to biologics, so we know like biological product, we defined biological product, source of biological products and all those. So, according to the set norms, biological products are diverse category of products that are generally large complex biomolecules, and they are produced through biotechnology such as microorganism, plant cell, animal, and often more difficult to produce than a small molecule, produce and characterize, right, than a small molecule. So, code of regulation of biological products. So, the biological products means virus, therapeutic serum, toxins, antitoxin, vaccine, blood components and its derivative, allergic products, protein and analogous system. They are various used for application of prevention, treatment, cure of disease. Those are biological products, right, and they are used for diagnostic, prevent and medical condition of various products. So, why they are difficult, right? So, look at the size of a biological product versus a small molecule drug. Traditional drug, small molecule drug, typically even if you take a small molecule drug, the atoms that are there or the molecular weight of this drug is 600 Dalton, right. You require few steps of chemical synthesis and you have these drugs, right. So, typically small molecule drug, medicinal chemists join hands with organic chemists, they synthesize this molecule. ये भी उतना आसान नहीं होता है because stereochemistry has to be maintained, that is also difficult, but one can synthesize this kind of drug because their size is small. Now, compare that with a biological drug. Any biological drug starting from peptide to bigger protein, the size is different, like very large. So, like a Herceptin, it is a breast cancer drug, monoclonal antibody, 185 kilo Dalton, 600 Dalton versus 185 kilo Dalton. Now, we have to produce this protein, although we are producing in live organism or the cell, like first of all this kind of protein cannot be produced in bacteria. So, it has to be produced in human cell. Their yield, their growth media, their temperature, their bond formation, many things can change. Therefore, they are very comparatively quite challenging and difficult. Anything can go wrong at any time in point, pH गड़बड़ हो सकता है, टेम्परेचर गड़बड़ हो सकता है, कंटामिनेंट्स आ सकते हैं, कुछ भी गड़बड़ हो सकता है, मीडिया में कुछ उल्टा सीधा चला गया, प्रोटीन सही नहीं बना. So, first of all it is done by organism or a cell, yield can vary, the product can vary, and therefore, every time you produce it, you have to characterize that whether we are getting the right kind of molecules. And characterization also requires involved, because it is coming from biotechnology process, it requires involved techniques, very high resolution techniques for characterization. Small molecule 600 Dalton है, कुछ experiment कर लोगे IR NMR तो हो जाएगा, इसमें उतने से काम नहीं चल रहा है. You have to do lot more experiment in details to find it out whether we are going with a correct molecule or not. So, there are many types of biological product as we discussed, therapeutic proteins like filgratism, monoclonal antibody, vaccines and all those, and that is what it takes longer and difficulty to get such biological product done. Okay. A little more detail about biological products as we discussed, allergens. So, allergen, like you know, many people have allergies for something, pollen allergen, it is very common. So, allergens are basically they are used to treat allergy in different people. So, allergen extract, it can be patch test, antigen skin test, many of these what kind of allergy people have, they are used for diagnosis as well as for treatment. Blood product, it is a very small biological product. Where blood product are used? Blood product, एक तो सबसे पहले खून चढ़ाने में काम आता है, होल ब्लड, राइट. प्लाज्मा थेरेपी में भी काम आता है, और कहाँ blood products has a tremendous, there are clotting, right. Clotting is done by blood product, right. Blood product अपने आप से clot हो जाता है. Many people have not sufficient clotting agent. So, either you have to fuse it or make some drug which is there. Our problem with our country manifold. कुछ साल पहले तक हम ब्लड प्रोडक्ट भी अपने यहाँ नहीं बनाते थे, सब मंगाते थे. आदमी ऑपरेशन थिएटर पे में पड़ा हुआ है ऑपरेशन टेबल पर, और उसके लिए एसेंशियल ब्लड प्रोडक्ट हम इंपोर्ट किए जा रहे हैं. कैसे चलेगा? ब्लड प्रोडक्ट्स basically blood or blood component or blood bank device, blood screening test, many other things, even small fragmentation of the blood, all will be called all can be classified into blood and blood product category. Now, another biological product is cell and gene therapy. Details will come as we move, but gene therapy, gene based treatment. Can you give an example of gene based treatment? Very nice. So, recently one drug got approved for gene editing technique, CRISPR-Cas9. Any of you follow the science, Jennifer Doudna has developed and she got the Nobel Prize for gene editing technology. So, now this drug has been developed which will cut the unnecessary gene which will develop sickle cell anemia, and that is a gene therapy. Or हम आगे पढ़ने वाले, many drugs are developed being AAV virus gene therapy, gene packaging agency. Cell therapy, stem cell therapy is used. So, many of again comes into biologics. Tissue and tissue product, graft, bone is a big problem, right. You need to have some patch that can grow it, otherwise पता नहीं क्या क्या उधर हो जाएगा. स्किन खुला है, all sorts of organism can go, it can be a major problem. So, bone, skin, ligament, tendon, smell, whatever, heart valve, all these one can call into tissue and tissue product. Vaccines, now we are बचपन से वैक्सीन लगने लगती है, so all of us know vaccine develops immunity against any pathogen like which is attacked. Then xenotransplantation, right, organ वगैरह लगा सकते हैं आजकल, कोई भी टूटा है तो सुअर का हड्डी लगा दिया ह्यूमन में, so this kind of xenotransplantation. So, all these comes into biological product, right. So, going back, so what is integrated drug development program? For a say biological product or a say biological product, protein based drug. So, we start from first you have to design a clone. Say I am developing a protein based drug or monoclonal antibody does. So, first process is going to be clone generation, process optimization and doing the analysis. Why clone? Why clone optimization? Is it required clone optimization? Yeah, we are going to say synthesize, or you want to get a human protein done in other organism. You have to codon optimize, right, so that we can produce more and more and correctly folded form. जैसे मान लो डाइसल्फाइड बॉन्ड हमारे प्रोटीन में बनता है, these are called say or PTMs, post translational modified product बनता है. Many proteins has a post translational modification. Now, bacteria is not suited for this, isn't it? तो छोटा ऑर्गेनिज्म है बेचारा, अपने काम लायक प्रोटीन बना लेता है. हमारे काम लायक प्रोटीन बनाए जरूरी तो नहीं है ना, तो उसको हमें ट्रेन करना पड़ता है, उसको हमें वो राइट काइंड ऑफ चीज़ें देनी पड़ेंगी, देन ओनली इट कैन मेक इट. So, for that, even for your CHO cell, which is commonly used for making of big therapeutics protein, first and most important thing, clone development. You develop clone where the probability of getting the right kind of protein is maximum and getting any sorts of deletion, truncation, wrong bond formation is not right. So, that is a clone generation, and you develop the process, analyze at every step, so that we have a right kind of. So, and then again toxicology, clinical study, this you can do in our lab, easily a smaller setup. But suppose I have to do now mass manufacturing, ठीक है, लैब में तो 2-4 लीटर हम कर लेंगे, mass manufacturing we cannot do in our lab, right? So this has to be done by Serum Institute of India like company, right? So the research lab Oxford University can make it, IIT Bombay can make it, but to make it to large scale it has to be done either in collaboration or you transfer the technology to them so that they can mass manufacture it, and then together you can do regulatory filing, post launch effect. So intellectual property, regulatory, business development operation. ड्रग तो बना लिए, बेचना भी है ना, पैसे जो $3 बिलियन या $5 बिलियन लगा है तो आना चाहिए वापस. So once you file and got approved, then marketing comes. Scientist का काम खत्म, businessman का काम चालू. It has to be sold, money has to be recovered, people should be, so all sorts of people will be involved in that post approval process, right? Business development, operation, transportation, many things comes into logistic, and that is a whole integrated drug development program. So सोचो कहाँ कहाँ opportunity है, साइंस करने से लेकर, रेगुलेटरी से लेकर, मार्केटिंग से लेकर, एवरीवेयर इट्स ए ह्यूज सेट ऑफ पीपल. एंड जनरली टू मेक लाइफ इजी फॉर इंडस्ट्री, अपने मन के अनुसार सब लोग ना करें, एक रेगुलेटेड वे में करें. FDA हैज गिवन all those guidelines for pharmaceutical development. There are many things you can do, the critical quality attribute, design, risk analysis and all those, right. So that's how one goes it. So let's take, we are going for a biopharmaceutical company. So what will be the organization of biopharmaceutical company? Now you fairly understood it, what all it takes, right. So this is the idea of, or this should be this constituents of biopharmaceutical company, organization structure of a company. So as I said, the first and initial work is done by the R&D guys, research and development guys who develop these clone process, and then once everything is very well optimized, abhi they transfer that knowledge to manufacturing. मैन्युफैक्चरिंग में ज़्यादा कुछ R&D करने का नहीं है. अभी 1 लीटर से बना रहे थे उसको 5 लीटर से, 100 लीटर से, 1000 लीटर से, 5000 लीटर से वैसे बनाना. So that's manufacturing. It has to be fairly automated, less human intervention, not so many parameters changed and all those. So those comes into manufacturing plant. Now you manufacture, but somebody has to finance it. So finance will control कितना पैसा जा रहा है, कितना पैसा आ रहा है. So finance will be there. And once everything is done, your drug बन गया, approve भी हो गया, तो marketing will be there. So marketing people comes and then they start selling them. Now in R&D, what all involves? Process development. You develop a process, do the toxicology, do the analytical study, the pharmacology. In the process development, you even do the clinical test, 1, 2, 3 जो मैंने बोला. And that's the, and then because all those will be controlled by this regulatory guys who has to license or coordinate with the regulatory agency to submit these drugs. They also file the intellectual property because you want to also patent your idea, process, whatever, so that you can recover the money that you have invested. And somebody requires a business development who can sell it, sell the drug, right. So वो तुम किसी डॉक्टर के पास जाते हो तो कोट टाई वाले कुछ लोग घूमते रहते हैं ना, right. So they visit, what are these called? MR ना, medical representative. So they go and visit doctor and develop the business, right. So इन सब के टॉप पर कौन बैठा होता है? CEO, right, who coordinate with this. So all these people put the effort and कुछ अगर गड़बड़ हुआ, कुछ सब कुछ अच्छा हुआ तो नाम भी CEO का होगा, और एक भी ड्रग फेल हुआ तो गर्दन किसकी फंसेगी और जेल कौन जाएगा? CEO, because he is the responsible for coordination and everything that is going on, right. So, process development of any drug. So, yeah, so process development, you know, this is the like a doing R&D, always we do R&D on a small scale. There are lots of exploratory stuff are there, we do R&D, there is a high chance of failure, some chance of success and all those. But once the R&D is done, now your core technology has to be developed so that you can scale it up with less cost. Production always requires minimum investment, maximum output. So essentially, and this is very well suited for like all the R&D तो केमिस्ट, फिजिसिस्ट, बायोलॉजिस्ट, AI वाले सब कुछ करते हैं, राइट. लेकिन जब इसको स्केल अप करना होता है तो अपने केमिकल इंजीनियर्स टाइप, एटलीस्ट बायोफार्मास्यूटिकल में केमिकल इंजीनियर्स आते हैं, राइट. केमिकल इंजीनियर्स का, यू आर फ्रॉम केमिकल इंजीनियरिंग, राइट. सो व्हाट इज द रोल ऑफ नाउ केमिकल इंजीनियर्स in process development? Can you list some?
Sir, as you already mentioned, one of the thing is when we do research, we work in very small quantity.
Small scale, yes.
When we go to a plant scale, many aspects change in it. So how to bring it, I mean, what all parameters to be adjusted so that it can be, the lab conditions can be replicated. That is where it could be kinetics, it could be some heat effects that come in, some mass transfer.
Mass transfer has to be done, yes.
So that all need to be optimized so that we can replicate the lab conditions.
Agree, very nice. Few of the things that I can even add it, like we always grow our bacteria or cell or something on some media, right. So it preferably eats something, preferably leaves something. So in this process optimization, you want to see कि सेल ने क्या क्या जल्दी जल्दी कंज्यूम कर दिया, कैन आई रिप्लेस ओनली दोज थिंग्स, राइट. इफ द रिक्वायर्ड थिंग्स आर गेटिंग रिप्लेनिश्ड, it affects the growth. So, or just depleted in the media, that cell now does not grow that fast we want, and for high production we want to grow it faster and faster. So, feed batch analysis, feed media analysis, spent media analysis, the oxygen level, dissolved oxygen level, pH, everything has to be tested and regulated perfectly, and chemical engineering guys, they learn all these. And when this comes into these kind of plants to be operated, the mechanical engineer, chemical engineer, like a design engineer, all of these guys pitch in there to scale it up. So, they develop the expression technology, the cell culture, microbial process technology, purification, because now हमारे लैब में इतना बड़ा कॉलम लेते हैं, 1-2 ml प्रोटीन प्यूरिफाई करते हैं एंड देन वी आर हैप्पी, राइट. बहुत प्रोटीन आ गया, बट नाउ यहाँ बहुत प्रोटीन का मतलब लीटर्स में प्रोटीन आना चाहिए, राइट. सो प्यूरिफिकेशन लॉन्ग कॉलम हैज टू बी रन, वेरी लॉन्ग कॉलम, दोज आर डेवलप. देन यू हैव टू लाइक बैच टू बैच वेरिएशन नहीं होना चाहिए. ड्रग यू आर मेकिंग, राइट. चूर्ण वी आर नॉट मेकिंग, ड्रग वी आर मेकिंग. So, in drug batch to batch variation should not be there. So, it has to be each batch or each type of product that is coming has to be thoroughly analyzed, and therefore, one has to develop analytical technology or formulation technology. So, once the proteins are made and it looks fine, it has to be stable for years, right. So, many drugs are made, they have to be transported against like all the odds. There can be agitation related problem, it can be heat related problem, it can be various problem. ट्रांसपोर्ट, क्योंकि बेंगलोर में ड्रग बनेगी, बेंगलोर में ही कंज्यूम तो नहीं होने वाला है ना, ये तो अरुणाचल प्रदेश के तवांग में भी जा सकता है, राइट. सो ट्रांसपोर्टेशन हैज टू बी देयर, टेम्परेचर कैन वेरी एंड ऑल दोज. सो फॉर कीपिंग द ड्रग इन द सेम स्ट्रक्चर, the one dedicated team of the people works on the formulation technology. How you formulate so that protein remains stable in that condition for months to year without any change. There is shelf life. हमने कोई भी ड्रग खरीदा, उसके एंड में कोई भी बायोलॉजिकल ड्रग भी देख लो, उसके पीछे में एक इंफॉर्मेशन होता है और एटलीस्ट एक चीज़ तो हर ड्रग में कब मैन्युफैक्चर हुआ, कब एक्सपायर होगा, माने उसका सेल्फ लाइफ टेस्ट किया गया है कि इतने दिन तक स्टेबल रहेगा. So those are coming from the formulation technology and then people develop it. So generally where do we grow our bacterial protein? So this is the clone development expression vector, we put it our gene of interest, and this we do in molecular biology and grow protein in various types of cells. The most easy and cost effective is bacteria, but it has its own limitation. So you grow in bacteria. The other can be the eukaryotic systems like insects, yeast, and finally many proteins are grown in mammalian cells. So that these are the expression vectors or cell line that is used, and this is kind of a huge fermentation and purification column that typically is used in any industry for enhancing the volume and making it more volume for doing this. And after this all the process, finally you develop a drug which has to be sold. So once this everything is done, you fill in the ampules or whatever, like whatever the, what is this called? Injectable. So it's a ampules or pen, some kind of pen or whatever this is called, a small vials, yeah, vials. You fill it, seal it, label it properly, have the all drug information, and now it is shipped to the market, and that is basically used. So this is the integrative drug development program that goes through for developing any drug. Any question up to this point? हाँ बोलो.
पॉपुलेशन कैसे डिसाइड करते हैं क्लिनिकल ट्रायल के लिए, कहाँ से पॉपुलेशन सेलेक्ट करते हैं?
कहाँ से पॉपुलेशन माने इनरोल करना पड़ता है ना. जैसे मान लो हमने ड्रग ये एक स्पेसिफिक मान लो इंडिया के लिए हमने बनाया है इंडियन, सो यू हैव टू मिक्स एंड मैच विथ द पॉपुलेशन व्हिच गोज इन क्लिनिकल ट्रायल. तो देखोगे कई सारे हॉस्पिटल को उसमें इनरोल करते हैं. ये नहीं कि मान लो कि ड्रग हमने बनाया और केवल महाराष्ट्र में हमने टेस्ट कर लिया तो काम हो गया. यू हैव टू इनरोल डिफरेंट हॉस्पिटल्स हु विल बी रिक्रूटिंग दिस पेशेंट्स and administering the drug, looking at the effect of the drug, कुछ भी adverse effect आया, it should be equipped to treat immediately. So this clinical testing should be done in wide population, like wide population means different ethnicity, yeah, sample size should be diverse. करते हैं तो ज़रूर हैं, क्योंकि उधर ड्रग टेस्ट करना बहुत एक्सपेंसिव है, तो इधर सस्ते में करने की कोशिश करते हैं. अफ्रीका, इंडिया, ये सब लो इनकम ग्रुप में दे डू इट, बट एसेंशियली इट शुड बी डन ऑन द सेम पॉपुलेशन दैट इज देयर. सो यू नो ऑलवेज देयर इज ए प्रॉब्लम ऑफ सच काइंड ऑफ अनएथिकल प्रैक्टिस, आई वुड से, बट that is already like that is there. I cannot say it does not happen, but should be chosen a diverse set of people.
जो जेनेरिक और बायोसिमिलर ड्रग्स हैं, तो पेटेंट के लिए 100% सिमिलर होना चाहिए उन दोनों का कि कुछ
हाँ, देखो मैं पहले थोड़ा बहुत जेनेरिक का दे देता हूँ. जेनेरिक की एक गाइडलाइन अभी में इस्टेब्लिश कर दिया कि व्हाट इज़ द एपीआई कंटेंट शुड बी देयर एंड ऑल दोज. सो इट शुड बी लाइक दिस गाय सुब्रता हैज डन ही इज डूइंग ए प्रोजेक्ट. So generally the API content should be 90 to 110%. So if that variations is matching, they call it, it's a generic drug. For biosimilar drug, biosimilar actually you know Obama signed the Biosimilar Act. It's not very long, 2009 probably he signed the Biosimilar Act. Obama कब president थे? Biden से पहले Trump, right? Trump से पहले Obama. So he was in 2 terms. So not long back biosimilar started. So Biosimilar Act is not very old, but what has been current practice of Biosimilar Act is that it has to be robustly matching analytically, structurally, like they call it HOS first has to be established, higher order structure has to be established, all the critical quality attributes should match. So if you do this robustly, you do not have to do so much clinical trial, only a relevant set of people, a small set of people you have to do clinical trial, and then biosimilar can be granted, right. but a new drug, a new biologics drug, extensive clinical trial required. This probably will come and explain more in detail difference between biosimilar and a new biologics drug.
बायोसिमिलर में सिर्फ एपीआई को ही कंसीडर किया जाता है मतलब पेटेंट पर्पस या फॉर्मुलेशन
बायोसिमिलर तो पेटेंट नहीं ना, पेटेंट ऑफ पेटेंट ड्रग है ये। हाँ,
एक्सिपिएंट्स को भी कंसीडर किया जाता
नहीं, एक्सिपिएंट्स मैच करना जरूरी नहीं, क्योंकि कोई भी फार्मा कंपनी अपनी सारी सीक्रेट नहीं बताएगी ना। You know your molecule, what is active pharmaceutical ingredient, rest you have to find yourself, and that is how biosimilar development also takes time and money. It is not that easy that a click mein ho gaya. You know the active pharmaceutical ingredient, you can express that or purify or whatever, synthesize that active pharmaceutical ingredient, rest formulation strategy and all those you have to develop your own.
And that mixture will be patented design patent.
The API?
API along with the excipients.
Probably not, I think, because there is nothing new, right? You develop, probably the process and all those maybe can be patented, but new drug is not, cannot be patented, because this is not a new drug. You can say process of optimization and stabilization. I am not sure, but we can discuss with a more expert. Process probably is a new, but whole package is not new, it is already approved. Right. So, in USA there is a branch called CDER, this branch of FDA that oversees drug, that is called Center of Drug Evaluation and Research. Basically this branch looks at the safety and effectiveness and this has everyone in that, a physician, a chemist, pharmacologist and other scientists. They are part of this regulatory team who looks at the data very, very carefully. So, there is a like a robust set of people who evaluate this data, and yeah. So, when the new biologics you are coming up which can treat a disorder or disease or diagnose some pattern, the first thing you do according to the act, whatever US Act 351, you must apply something called BLA, biological license agreement, and this biologics drug they must comply with Food Drug and Safety Administration, that something called 21 CFR point, and they have to be manufactured in something called CGMP, that is overseas by this agency called CDER. What is few of the term that I mentioned? What is CGMP? Good manufacturing. C क्या है? ये नहीं कि 17वीं शताब्दी में जो गुड मैन्युफैक्चरिंग प्रैक्टिस है उसको आज भी यूज कर सकते हैं. So, what is like so it has to be done in a certain regulated manner to be done. So, history of biologics regulation, right, earlier there was no biologics regulation, but over like started from 1938, there are lots of like there is a history that maybe sometime I can share with you. Some problem happened in District of Columbia where people got adverse effect and they were dying, and then senator complained, and then these people sat together, made a really robust regulation way, and that for over the years initially it was done by NIH of USA, later on this new organization called FDA came, and then every drug now whatever has been developed, it has to go through these regulatory process. For USA and also for Europe it is a EMA, and for our country it is a CDSCO. Every country has their own way of doing. So, process of drug approval, the investigational new drug process for biologics, it should be 21 CFR 321. These are all documents. You probably once you enter into this process you will remember this, but at the moment it's okay if you even if you don't remember. So, FDA regulates the clinical study at least in USA, and why USA? Because they have a set protocol. ठीक है, EMA and USA has a set protocol. बहुत सारे दुनिया के देश अभी भी फॉलो कर रहे हैं वही सब. उनका 1938 में स्टार्ट हुआ, राइट. सो इट्स वेल टेस्टेड, वेल रोबस्ट. Those are regulated market, and there are many unregulated market जहाँ पर उतनी stringency नहीं है. So USA में drug बेचना, that's why all the Indian company, many of them are trying to crack to US market. एक बार US market में approval मिल गया यानी बाकी जगह आसानी से मिल जाएगा. So that's how they target. There are lots of time, energy and everything goes in getting it because they have a set protocol. So biologics is 21 CFR and INDA already we explained to you. So FDA regulates clinical study in USA, and unapproved drug and biologics must be conducted under investigational new drug. So they are not called new drug application, this is called investigational new drug. So end up when you find it must contain like administrative information, preclinical result, previous human experience with the drug and clinical protocol. And finally, the analytics and product quality analysis of drug development has to be done at the screening stage, characterization stage and release stage. So all the data that has to be given for FDA and then finally the guidelines for FDA is prescribed, which has to be followed for preclinical safety evaluation for biotechnology pharmaceutical. So these are the typical points that has to be taken care once you go for new drug application. This I will stop it here and we can take it from in the next class how we go to forward from here. So probably you got an idea what all it takes. Now last thing that I want to discuss, since I already mentioned you and you guys always are interested, what are options for you? So look at the biotechnology career options, right. So chemist already required, biologist required, chemical engineers required, physicist required, medicine guy, pharmacy guy, mathematics and statistics, because clinical data has to be done statistically significant and all. Computer science and engineering. Now AI is coming in drug discovery. You require a person who understands the biology and you require a person who understands the computer science, AI and all those. So those are also required. Other than that, a mechanical engineer, because you are making huge machines, chemical engineer for increasing the production, right. Then from medical side, physiology person is required, physician is required. From biology, you require a biochemist, a molecular biologist, bioinformatics, the genesis like who can do the molecular biology and gene synthesis and all this, chemist is required. Then other than these are all scientists and engineers. Other than this, management guy you require, marketing guy you require, right. The person who understands the law and regulatory laws, those are also required. The finance guy, economics guy, और जो दिखता है वो बिकता है, इसलिए दिखना भी अच्छा चाहिए. So you look at the logo design, the ampoules design, everything design has to be done. Graphic arts and designing is there, and scientists, whatever scientists say, it has to be written in a language that common people can also understand. So scientific writing guys are also required or whatever. So all these are like a career opportunity for a person who is entering into biotechnology, because it's a huge agency where different kind of expertise is required to run this. Okay. So with this, in the first class, in the second class I end, we will discuss this in the next class and starting with a new topic. Go ahead.
Promises the marketing team for pharmaceuticals in India where we don't really see ads for medicine.
You don't see ad?
In terms of a commercial, like USA where they have like advertisements for, let's say, a cardiac or a painkiller. Here we don't have that. Mostly it's generic.
Yeah, first of all it is generic, but nowadays lots of things are coming. थोड़ा हम लोग पीछे चलते हैं ना, तो इसलिए things are coming, not that aggressively. Now Amitabh Bachchan is giving ad for this vaccine called shingles, you know, he is giving, many people are giving some kind of, अपने बच्चों का वैक्सीन लगा लीजिए, ये कर लीजिए, वो कर लीजिए. So there are lots of ads are coming Not in that big way, but still it is.
I think isn't this government?
No, it is not a government, it is a private pharma agency are advertising for themselves. But many of these things, yeah, you are saying it is not done aggressively to the common people through through TV or whatever, newspapers and all those. Basically, when you go to hospitals and all those, you see lots of advertisement, right? When you are becoming a father, जो कुछ सालों में तुम बनोगे, so you will go to whatever the maternity home and all those. बहुत सारे ऐड आते हैं, अपने बच्चे का इम्ब्रायनिक कोड वो कर दीजिए, स्टेम सेल में फ्रीज कर दीजिए, ये करिए वो करिए, सब दी जाते हैं. हाँ, राइट राइट.
So, but
हाँ.
there's a problem with the advertisement of medicines. If you start advertising all the medicines, people won't go to the doctor. They take it without prescriptions as well.
No, so many of drugs are prescriptional drug, or yeah, that has to be done with this कि किस इंडिकेशन के लिए ड्रग है, कितना डोज में आना चाहिए. People, that's right also. So, but for few things it is being advertised which is preventive kind of or diagnostic kind of. Treatment has to be done in a controlled environment. अभी कुछ over the counter drugs हैं, you can just go and buy, right? In regulated market that is not right. In Germany, pregnancy kit cannot be, pregnancy testing kit cannot be just purchased. ठीक है, अपने यहाँ जाओ किसी भी काउंटर पर मिल जाता है. जर्मनी में डॉक्टर हैज टू राइट you want to test it, then only you can buy it. So, there are different kind of regulation in different country which allows either to be sold or not to be sold. Okay, thank you. See you in the next class.